How are you? Hello, Conrado.
Hi.
How are you? Fine. Thanks for joining us. Really nice of you to present.
Thank you. Thank you for the invitation.
We're quite excited by your publication. There's some overlap with analysis that people have done here on synaptic data, and I think we see similar results. And so it would just make sense to fold you in and have your perspective and share and see also your results.
Thank you. I'm very excited also to discuss about it. And let me know when.
Let's give 1 or 2 minutes because people. Sometimes I'm myself late.
Jerome is always late. Jerome, could you share the paper in the chat, maybe?
I believe I sent it via emails. I'll send it again.
I think I don't know if it's only me, but Conrado, I can hardly hear you.
If I'm speaking now, do you hear me?
It's I don't know. It's for me. It's very low. How's it for you, Jerome? Or
Yeah, it's a little bit muted.
Oh,
I wonder.
Now it's better.
Oh, that's better.
Yes. No, it's I have another. Yeah, it was very the microphone was very far away, but now it's fine.
No,
yes,
I need
no,
to.
I'm using my AirPods now, so yes.
Fantastic.
Yeah,
so
thanks for noticing.
Why don't we get started?
Yeah, I'm gonna share the presentation. Just a second.
So let me give you a couple of words of introduction. So first, as usual, this meeting is going to be recorded and we plan to post it on YouTube. So if you have any concern with the content, please let us know at the end and we can make corrections.
Yeah, no, it's not a problem. Yes.
And so today our agenda is a little different from usual. So we're going to discuss a publication, a recent bioRxiv publication pushed out by Dr. Bosman group and collaborators. For context, we were doing some analysis on SLAP2 data and found some interesting overlap with that really recent publication. And so we just decided to invite Conrado, who kindly agreed to present. He is from Netherlands, currently in Amsterdam. And so we are really looking forward to your presentation, Conrado, and
thank you very much. Thank you. I'm glad to be here. Exactly as I say, I'm Conrado Bosman from the Swammerdam Institute for Life Sciences, the University of Amsterdam, in the Netherlands, originally from Chile, Latin America, but now long time living here. And I'm happy to share my work, my recent work on how predictive context can reshape orientation coding in B1. And as you already mentioned, this is based on this. What I'm going to present today is essentially this recent preprint that it's in bioRxiv. So let me give some words of introduction to give a little bit of context. And the first slide what I want to share with you is this one that probably everyone is familiar with, is the Hubel and Wiesel iconic stimulus orientation paradigm used in cats and then monkeys, late 50s and during the 60s, in which they discovered these cells that respond selectively to specific edge orientations. And the bottom line of all these experiments and the classical view is that this orientation preference is considered pretty stable across conditions. It arrives early in development and it's there, and we take this as a given. And this, of course, set the baseline assumptions from all the work that has been done in visual perception. However, we also know that the processing or the cortical coding isn't fixed. It depends on context. And in terms of context, another iconic work is the model of Ramon-Ballard, which essentially shows the idea of the predictive coding, in which we said that there is some sort of a model of the nature, and what the animal or the individual has is some sort of an input in which there is some sort of comparison with this model in the forms of prediction, in the form of feedback. And if there's an error signal or some deviation from that model, it goes the feed-forward connections. And also, they not only show this as a model that occurs in one specific level, but then there is some sort of hierarchical of these signals in order to this sort of upgrading and updating model every time that there is some sort of important perception for the animal. And this model explained quite well context-dependent modulation of visual responses. In the same Ramon-Ballard paper, they have this sort of example in which they have here the response of a particular cell in two different conditions, one with an homogeneous random textures and other with a more random wise. And here it says it shows that how the response decrease the number of the spiking rate of the responses lower when there is an homogeneous random textures compared with one that is random. So this then set the context of this study. One is trying to see this idea of representation in terms of orientations, and the other is the idea of study this under context effects. And you also probably are very familiar with the way that this is that you can study context effects, and this is using the Oddvall paradigm. In the Oddvall paradigm here, that has been well studied in auditory cortex, less so in visual cortex, but we know how and people from this consortium has contributed with excellent papers on the Oddvall the utilization of Oddvall paradigm in visual perception. So, but the original formulation, as you well known, is that you have a presentation in tandem of standard stimuli, and then you have a deviant one. And what it is possible to observe is that in this case, taking for example ERP responses, you see a change of the waveforms, and when there is a subtraction, you observe the classical mismatch negativity response. Now, regarding what the circuit component and how that happens in B1, there is, as I said before, a very active field of research, and we know much more now about the laminar and feature-specific mechanism, but still the dynamics needs to be clarified. So, in order to do that, we set this study, and we asked the question whether prediction can reshape feature preference in B1. So, can, as we said before, it is assumed that the orientation preferences are fixed, but the predictive coding can suggest that they could alter some feature selectivity. And the two aims of this study was to test whether there is some there could be some changes in orientation preference, if there's any changes on stimulus predictability or change in the context, and also the second aim of this study was to examine the effects across cortical layers. And how we approach it, we did with an Oddvall paradigm, but we use this modern formulation of the odal paradigm used also by other researchers here, in which we have a presentation of standards, and you can see here, in this case was a particular orientation, and there was a number of standards. I think it was 7 standards, and then we presented a deviant, which was an orientation orthogonal to that, and those was defined the two conditions, the standards and the deviant. But of course, if you want to disengage the effect of mismatch negativity, but also the effects of deviant detection, you need to have an additional control, and that is what we use what is called the many standards, in which you present the controls, in this case, this same orientations as the deviant in a sequence of many standards. Of course, we did all the paradigm. We did it. We counterbalance, and we use the orientation on standard sequences and also the deviant sequence, as doing by others also. This was a passive presentation of this this stimuli for 500 milliseconds. And the animals were awake, and in order to study the effects through cortical layers, we use laminar electrophysiology, in particular some neuronexus probes, 32 electrodes each implanted in P1. We present these whole field moving ratings, and as I said, it was a passive presentation. So what we did first was to try to see whether we can really get signals, meaningful signals from these presentations, and that is. So the very first thing that we did was to pull all the ERP response together across all the electrodes, and then calculate an average, and that then we observed then the response of the control, the standards, and the deviant, as you can see here. And then, of course, we calculated the contrast. In this case, the stimulus-specific adaptation, the mismatch negativity, and the deviant detection, which is the subtraction of the deviant to the control. And see that we observed effects here, robust effects on stimulus-specific adaptations, on mismatch negativity, and in deviant detections in 2 temporal domains. So the first 200 milliseconds after the stimulus presentation, what they call the early latency, and after that from 200 to 400 milliseconds late latency. And the deviant detection was observed in the early stage, not so much in the late one. We observed that also we focused on the spikes, and this is one example of one of the cells that we recorded, and here we see the response pulled across all the electrodes and all the neurons that we isolated, and here you see the firing rate to the 3 conditions, and here the 3 contrasts. And what we observed here, the difference is that the deviant detections can be seen at early latencies and also late latencies. So this preliminary approach confirms that the paradigm can elicit responses both at synaptic level, but also at spiking level, synaptic integration and spiking level, and the results are mostly similar to what has been observed before. Now, in order to study the layers specific dynamics, what we did was to the very classical approach. So we combine anatomical localization of the electrodes together with layers, and we got the increase of multi-unit activity, which is usually been characterized as part of the infragranular layers, and we use current source density analysis also to identify the sinks and the source change, which shows the granular layer. And with that, then with this characterization, then we classify or we sorted our neurons and our electrodes in these putative 3 layers. And here you see the responses at the level of the ERPs. And what we observed then was some that you can focus here on. So you have supragranular and granular and infragranular, the early and later response on contrast. And we observed early ERP responses with robust stimulus-specific adaptation, mismatch negativity, and deviant detections, and that was the stimulus-specific adaptation and mismatch negativity across all layers, but deviant detection was significant only in supragranular layers during the early phase. In the late phase, the response was observed. So we didn't observe the deviant detections at the level of the ERP. We only observed these stimulus-specific adaptations during infragranular layers. However, when we study the spikes, and so that can be summarized, and we found an early deviant detection in supragranular layer 2-3, and late stimulus-specific adaptations in infragranular layer. When we study the spiking responses, and here I must say that while we get some significant effects, our statistical power for the mixed models were only sufficiently strong for infragranular layers. But we observed that tendency to see some effects for early increases for deviant and controlled stimulins in supragranular neurons. But the most important response we observed at the infragranular layers, and then it was when we observed this late increase in firing rates for deviant stimuli. Which it's a bit of a parallel what you observe in the ERP responses for the stimulus-specific adaptation in these layers. So these patterns then suggest that the deviance detection starts in superficial layers and is expressed later in deep layers, and so both at synaptic and spiking levels, and in a way. And we can discuss later how that it compared to other similar studies in the literature. But this is what we observed. We wanted to then to see whether is there any. We're trying to now go into the not only on the dynamics of the responses, but and how these responses can alter the representational context of the cell. So we use a population coding approach to decode and characterize whether these particular populations or the particular latencies can accurately decode the stimulus orientations from the population spiking activity. And what you have here is the decoding accuracy for the 3 conditions during baseline. And here you have for during stimulations, and you see in this plot that one side mirrors the early effects and the other side the late effects. And this is the delta decoding accuracy, the subtraction between the baseline and the stimulation period. And what I would like to first is to mention that we found that the decoding accuracy was during baseline. We found that we were able to decode the deviant and standard stimuli, not the control, the many standard control. and we can discuss why that was where we found this. But the important thing here is that we found that the decoding accuracy increases for the deviant conditions at early latencies compared to the other ones. We got strong decoding accuracy during late latencies for all conditions, but the important thing here was to see that the deviant increased during the early window. So then that tells us that effectively the deviant improves the early orientation discrimination at the population level, but of course the coding accuracy is just it doesn't tell you how the population activity change across conditions. So to do that, you needed to go a little bit deep, and with that in mind, we decided to do an analysis of the variance of the activity and to study the variance, how the population move across the conditions. We use these metrics of the Manhattan distance, which help us to quantify the dissimilarity in the population activity. It's just simply that for each neuron, you look at the firing rates to two orientations, let's say horizontal and vertical. You took the absolute difference, and then you sum those difference. And what is interesting of this approach is that it's less it is less affected by outliers. And here in this matrix, you can see all the conditions and all the latencies together, and the population distance variance of one to the other. So here, for example, you see the when you in this case, for example, the baselines. These are the three baselines comparisons that we have. One to the control, to the standard, to the deviant. And what you can see here is that before the stimulation, you see the whole Manhattan's distance and the count of cells that have different Manhattan's distance quantities. You see here that, oh yeah, there is a question. Yeah, Sid, please.
Yeah, just really quick. So when you say variance, is that trial by trial variance? What variance is that?
Yes, it's trial by trial variance. Yes.
Okay, and so why did you choose Manhattan distance over, let's say, Euclidean distance?
Yeah, well, the decision of use the Manhattan distance was instead of the Euclidean is we find that it's less sensitive to outliers, and yeah, you can interpret it also as additive contributions per neuron. We did in a way. So that's what we decided to use the Manhattan distance instead of simply the Euclidean. But yeah, of course, Euclidean can also be used.
I find it interesting because my understanding of Manhattan distance is basically instead of going a direct line, you're assuming there's a grid, right? So that's why it's called Manhattan
yes,
distance.
Exactly. Yeah.
Okay.
Yeah.
So in a way, you're discretizing the data then, right? So you're assuming that the latent space, whatever you want to call it, that you're investigating has a discrete structure. It's like a Euclidean grid.
Yes, exactly. Yeah. Yes.
Not Euclidean. Yes. Sorry, Cartesian grid.
It's a Cartesian. It's a Cartesian because you go so you go in. You go in the. You don't go in one line. You go. Yeah. You follow this sort of matrix or this space that you get into which is calculated by the absolute differences between.
So really quick, by the way, it's beautiful work, by the way, and just trying to wrap my head around it. So why did you go after trial by trial variance? Arguably, that's the least important variable for the brain because it only gets one trial, right? So why did you go after that?
Yeah, I think we wanted just to have the most reliable characterization of the variance across all the cells. So that is what was calculated in that way. But I agree that it's also possible to calculate the variance as the variance of the cell in total, and then just group to group all the population together, and then just get one value. But yeah, I think it was a bit only, I must admit, it was a bit intuitively that we decided to, and we didn't explore all the possibilities to say, okay, we did it in this way, and it gave us these results, and yeah, we took consideration that the calculations were done.
Just so quick, maybe where I got hung up is that you showed this decoding, which is awesome, because I think that has been missing in the field so far. And then you said we're trying to find out what the decoder is using in a way, right? So we know there's some information. So, but isn't the decoder doing trial by trial? Like, how did you decode? Did you session by session, trial by trial? What did you decode exactly?
I will need to see the details on what we discussed, but I think the decoding was made in trial by trial. I think we tried to see whether we use this algorithm and then try to see whether the cell effectively can decode one condition or the other, and also that is also why we obtain in the decoder the significance for the standard and the deviant, because of course you have the only two options, so it's kind of it's always going to give you one option.
So I think, and correct me if I'm wrong, I think I follow the logic now. So you're saying there's additional information in oddball trials. Yes. So by showing that the variance is different is showing you, something is changing, and so that.
Exactly. Yes, something is changing there, and we wanted to.
Otherwise, if the variance, let's say, is.
Yeah, in a way.
Do you know if the variance goes up or if the variance? Do you know if the variance goes up or if it goes down for the oddballs? Do you know that?
Yeah, so this is what we have in this setup here. So you can see that because here, for example, you needed to see this is the part of the variance. So you compare baseline with deviant in the early deviant, and if you compare, for example, baseline with the standard, and that is what you have here. And then you see that the deviant. The deviant goes. So this is the deviant. So you see that the deviant is high. So there is a shift on the oddball compared to the standards. That's what we said, that the mismatch triggered a larger shift in the population state compared with the standard. And and also this is we're trying to point here what is creating this variance, or why we have this moving of the variance, and that is one of the things that we then trying to connect with the orientation selectivity. So we said, okay, if you, for example, take there is certain correlation of this shift of the variance with the orientation selectivity index, but of course you cannot use the orientation selectivity index to calculate orientation here, because in the deviant and the standard we only have two presentations. So that is what we create this idea of the orientation preference index. Then we try to see how the orientation preference index change in during different presentations or in a different context for the cells. And here, for example, you see two examples of two units. So in this case, for example, this unit has a larger orientation preference for that can be seen in the deviant horizontal and the standard horizontal, but you don't see it. You see that it changed in the control conditions. And more interestingly, this is a unit in which you see that there is a clear preference on the late latencies for the horizontal, but then it changed and you get it for the vertical. So when we collapse all the neurons that we have, we see that well, we have a significant number of cells that are not that doesn't show any preference. We have certain cells that they have a preference like it's fixed, but a number of those shows these flips between the conditions. And to show that more clearly, you can see here. So in these scatter plots, you have the orientation preference index from vertical to horizontal. And here you see the controls and the standard, the deviant, and compare with the conditions here, standard and deviant. And then you see here, because this is the switching area, you see we got a higher number of cells that, or a significant proportion of the cells that we recorded, and they have shown certain preference switch from one orientation to the other. And when we observe that, we try to see also the laminar distribution of that, and this is what we show here, in which then you see here that we observe this mostly on the infragranular cells during the late latencies. We wanted that to, of course, try to control whether to see that this is due to whatever random fluctuations or that it happens during any other presentation of the stimulus. So what we did then, because you have several standards, we compared the standards presented the first time with the second, the third, until with the seventh presentation of the standard with the deviant, and we only see those changes when you show the deviant, showing that a particular number of cells has this sort of flexibility. Not all the cells, but some of them. And then just to finalize, what we did was we tried to see whether this preference switching is informative or not of the orientation. And what we did here was just to use we have the decoding again with the preference switching neurons included, and in which we use a matched stable control, and we didn't see any difference on the decoding. So cells, the cells can do the decoding without any problem. We use this approach because we didn't have a big number of cells, but in the supplementary figure, we have also only the preference switching cells, and while the numbers are lower, that also shows that the switches, well, as we call it, encode as well as stable neurons also. So just to show you some now that I'm finishing here, the key takeaways is we found this orientation tuning in B1, the switch with the context. We found that this switching of the presentations is concentrating in extragranular layers. The population codes remain stable or can improve during the deviant conditions, and we also found that there is a timing of the deviant detections that go from the early supragranular to late infragranular. Of course, there are some limitations of this, and one is, I think, the problem that we are not studying preference, orientation preference at the classical way, because here we are using an index of the preference instead of the selectivity, and we have these space restrictions of the cells. But still, you can see some examples in which there are some dramatic changes in one cell from one orientation to the other. And of course, we don't have any manipulation here as in other studies that can show to distinguish better what are the local effects versus the feedback-driven effects. And yeah, of course, in other contexts, other than the Odwald paradigms, it can build different dynamics. This is something that also need to be explored in the future. And for me, it's important to say we didn't analyze any spectral or laminar interaction. Being myself very high affinity for study the spectrum in the LFP, we decided to let that from later. And with that, just I need to give acknowledgement in particular to the two PhD students that did the study, Tom Sickent, who did the recordings, and the paradigm, and Jeroen van Datselaar, who did most of the analysis that I'm showing here. And well, thank you for your attention. And yeah, if you have any question, I'm happy to continue the discussion. Thank you.
This is fantastic. I think there's a lot of directions that I think Alex said that the field was probably missing. I'm curious because you have this early versus late characterization. What do you think is the mechanisms that is causing those context switch? Does it involve certain inhibitory cell types, or do you envision other mechanisms?
Yeah, this is something that I don't know what would be the mechanics here. If there is some sort of inhibition of the cells that in certain conditions are giving to you a very restrictive orientation, which is a bit lax during the deviant detection, that could be one. But I'm not very. I think that could be one, and that would. The other is that there is some sort of feedback that is coming either from subcortical structures or higher areas, particularly because this occurs during the latencies instead of the early. That also can be, but unfortunately here we couldn't go show anything else than how the effect that this effect occurred in B1. It would be very nice to see how it happens. Yes.
Okay.
I have one question. So very, very interesting work. We found that when we look at the different blocks, control versus the deviant or standard conditions, that there's a bit of a transient in the responses. In particular, it seemed that it takes actually quite some time for responses to recover from the standard representation.
Mm-hmm.
We showed a lot of times, and I was wondering how long your blocks are, and whether some of these modulations of, that you see in the orientation preference index might be actually caused by essentially the sequence of blocks, and that some, let's say, adaptation effects bleed over into the other one or so.
Yeah, well, so for example, I'm seeing this as some sort of gravitational drift or some sort of variability that you can accumulate because you are presenting during several times this same presentation. But here I don't recall the exact number of blocks, but what we try to do in order to see whether there is some effect on this sort of slow accumulation of stimuli across time was to try to do this comparison between the standards, and in that case you don't take in consideration the block. You just simply we took in consideration one presentation versus the previous presentation, and then of the standards, and we didn't, and what we found is that the orientation preference index didn't change dramatically, except in a couple of neurons. However, when that presentation occurs during the deviant, or you have the last standard and the deviant, then is when you see these changes, and that occurs inside the block. So yeah. Also, we try to have so we presented the gratings for 500 milliseconds, but then there was a period up to 500 milliseconds in addition, in which there were no stimulations to try to avoid this sort of accumulation. So yeah, I would say, at least with that control, we feel that it's improbable that there is some sort of this slow drifting that could explain the result.
Alex?
Yeah, just two things really quick. I mean, one is, Conrado, you started with the Raoult and Ballard model, and this doesn't really go at all with it, right? So Raoult and Ballard would just say there's just some boost or some diminishment of activity overall. So this really adds a lot of complications. I just say, now, the other one, I don't know if you know, there's a long-standing debate in the field whether orientation columns are really orientation columns. So there's work in the cat where they try to go exactly perpendicular to Cortex. And if you look at the orientation tuning, it often falls apart in the infragranular layer. And the reason that a lot of people don't know about this work is that then there were later studies where it got dismissed. They said, well, maybe the electrode was oblique. But if you look at the actual, so they crossed from one orientation column to the other. But if you look at the actual data, it can't explain that, because what you see is that it's a very abrupt, exactly as you cross the layer 4 or 5 boundary, that's where it jumps. And it's often exactly what you see, that it flips to the opposite. And if you look very carefully in the study by, was it Tyrone Moore's lab, but they look very carefully using neuropixels at the orientation columns in the mouse. If you look carefully, you see it in that data too. They don't make a point of it. And so when we looked, what we found, and so that's why it might be interesting to you, is that we don't often see that jump in the orientation tuning, but so we also make sure that we believe we're perpendicular by looking at the receptive fields. And you can see receptive fields drift sometimes, and you're not perfectly across a column, but sometimes you have to accept the receptive fields exactly underneath each other. That's easier to do when you have small receptive fields, photophobia. And what we found more is that it's the variance of the orientation tuning that abruptly becomes larger in infragranular layers.
Interesting. Yeah.
So I'm wondering, since you're kind of going after, well, it's some neurons and not so much others, it's mostly in the infragranular layers, if you have some kind of more than just 2 orientations in your data, if you could go after the tuning curves, whether you could predict almost that the neurons that show you effect are some neurons that already have more, let's call it flexible orientation tuning, that maybe their variance is wider, something like that. Do you have something that allows you to get tuning curves, orientation tuning curves with these recordings?
Yeah, so the only so we need to use the many standard conditions, because then you have at least 7 different presentations, and then we can do orientation tuning curves and calculate the orientation selectivity index. I must admit that we didn't do this in the detailed way that but definitely would be interesting now that you mentioned to check now all the data and see it, the selectivity, just to take this many standard controls and see across the layers whether it shows that or not. Maybe it shows. One thing that we did was to, when we calculate the orientation selectivity index, to see and this is a figure for that I didn't show here, but it's in the supplementary figure. There is some sort of correlation, like it's a very gross correlation with the Manhattan distance, and yeah, it more or less it shows that there is some sort of shift. Whether it can be that effect that you mentioned, yeah, that is something that we will need to check. But I will be curious. Though this paper that you mentioned is from you, no? Or you did no?
No, we never publish it because it's such a dogma in the field. Yeah. That it's difficult to publish that. So these are papers from the 1980s, and but it's funny because my colleagues and I, we know about these papers, and so I remember that when we saw the Turing-Moore study, we were all going like this, and see, there it is again. So there's definitely something odd about the orientation tuning in the lower layers in V1, and so that is exactly why. Was like, oh, see, that's what you find. Technically speaking, what has been so influential was the Hubel and Wiesel model of how orientation tuning comes about, which is that it's hardwired from the LGN, right? So you have some circular LGN, and so you're showing, no, it can't be that. It must be software, not hardware. How could it change if this were all just because of the hardwiring, right? So this might explain layer 4 orientation tuning, but it becomes different in the infragranular layers.
Yeah.
So
I agree.
It's almost, it reminds me a little bit of, because I haven't seen this before. I don't, maybe other papers that show it. It reminds me of when they did attention studies in V4 and they showed that the receptive field can shift. The same idea that it's how, if it's all just convergence in the hardware, how can the software do that, right?
Yes, exactly.
And so no, if you plan an eye movement, your V4 receptive field shifts towards where the eye will land. So it's clearly more flexible than it's just the axons that are already existing. And so now you show that for V1.
Yeah, exactly. And in this particular context, but I agree with you that there is a studies that there is changes on the receptive fields also, and also the effects of attention. So yeah, I didn't want to go too much into that rabbit hole because I know that it will raise angry reviewers saying that. So we wanted to show it here and be very cautious about what it could be. But certainly, I agree with you that it cannot be that it's so rigid in this way.
Can you remind me? Have you looked at the orientation tuning of the early versus late phase? I think you did that in your plot.
Yes, we did. We have cells that so we compare orientation tuning or preference index at the early latencies, late latencies, and we see that in some cases, in some you see the switch at the early latencies and also switch at late latencies. The thing is that most of the switches that we observed were at later latencies, but we observed a significant proportion at early latencies too.
Because you could argue that, for instance, what could be going on is the sort of the inhibitory networks, the EI balance is very different between the early and late phase, and so you have a switch of and so the cells sort of control the input. The inhibitory cells control the inputs on the dendrite, so you could get a very different tuning depending on the state of the network. And so to get the full picture, we really need the inhibitory neurons, I might argue.
Yeah. Yes, I agree with you. And in this case, we just we have very not a large number of inhibitory neurons when we did the recordings, and for this particular analysis we let them out because the number was not significant. But definitively it is definitely in order to find whatever mechanistic explanations, the inhibitory neurons need to be considered here. Yeah.
So, Conrado, I don't want to toot my own horn, but I have a PNAS paper, pretty old. It's called Context-Dependent Perceptual Modulation. And in a way, it shows what you show for binocular rivalry. So we recorded an MT, and the results could also be interpreted that depending on which stimuli and which eye, it changes the orientation tuning, the direction tuning of MT neurons. Yeah, and so back then, again, it also seemed heretical because direction tuning should be hardwired, right? And so, and it was also only some neurons, not all neurons. And back then we didn't have the lamina information. So
I remember that paper. I should have been okay. Well, I'm gonna check it again, and certainly I'm gonna include it after the revisions, of course. Yeah, that because I think. I feel that I was a bit didn't want to go too much into, but yes, definitely would be interesting to revisit those. So thank you. Jorge.
You okay?
Yeah, sorry. I just wanted to jump in the conversation because Alex, what you mentioned about the hardwiring of LGNs is something that is I find it very interesting. I put in the link some modeling work that we just did recently, and I think it resonates with what you just said. We see a sharp decrease for orientation selectivity when we go from layer 4 to layer 5, and it is because of the yes. So it aligns well. It is because of the plasticity at the level of cortical level, not LGN. So I didn't know about this debate in the field, so I was quite happy to hear it from you. So just to say that.
Before we finish, Conrado, I want to mention that we have a lot of discussion on the GitHub forum that I linked, and so if you want to engage with some of the plots and so on, please feel free to jump in and debate with. For instance, I'm really curious to hear your opinion on some of Lucas' analysis that is looking into the context a little bit that I linked into the chat.
Thank you very much. I was just checking the chat and I saw this. I really open it now and yeah, thank you. I'm going to read it and if I have something to say meaningful, I will jump in. Thank you.
Any other questions for Conrado? Well, I think I'm going to have to read this paper again.
Yeah, well, I think
I'm going to put in the chat one of these papers. So the lab that found that first reported these differences between upper and lower layers was bioR. So I'm going to put this in the chat. And I just found the one paper really quick, but you can probably go from there. That there were several, and then there were papers that attacked that paper and for the time settled the debate. So here's the original paper by bioR that showed the differences between the supragranular and infragranular cells and how the tuning shifts all of a sudden.
Thank you.
Okay. So let's discuss briefly next week. So Conrado, we are in the middle of deploying experiments to study these effects. And so our goal, I mean, I'm sure you can look at the review that is on arXiv. Our goal at the moment is to finalize the stimulus design of an experiment where we have 4 different types of contexts to look at these effects. And we're planning to do recording with both neuropixel, like large-scale neuropixel recording and imaging. One of those contexts is a sensory motor oddball context that Georg Keller's lab has pioneered many years ago.
Yeah.
And so at the Allen Institute, we are just in the middle of doing this experiment. So last week, I started to pilot mice on the wheel. And so I think next week we should discuss those. We have started to put mice on the semi-VR system where the mouse is controlling the flow gradings with the disk. So this experiments are starting to work. I'm actually quite excited to see that the mice are engaging with the wheel very much when we turn this on. So I think next week we should discuss those so that we can finalize the few first experiment we will run on NeuroPixel with this context. What else should we discuss? And then Severin wanted us to finalize the targeting scheme for NeuroPixel. So I feel like next week we should have this discussion, probably unstructured, unless some of the people have things that they want to have a discussion next week.
So I can speak briefly on analyzing the new data, Jerome. We had a little bit of a discussion on that.
Yeah. Next
so
week.
The big one is that the second file might have an issue and might have to be pre-processed again.
Okay.
But I can try to make progress on the first version. I got responses and I shared the code. So yeah.
So we'll try to fix the issue you raise within the week, and I'll keep 10 minutes for you next week. Sounds good. Anyone else has things they want to raise? Well, thank you, Conrado. This was really nice. I really appreciate it from the group.
Thank you very much for the invitation. For me, it was also nice to have this discussion with you that are so much into these topics, and yeah, I feel very honored to be invited. Thank you.
And feel free to continue to engage this, and you can join the meeting next week if you want. It's really open to everyone.
Thank you very much. Thank you. I will. Thanks. Well, have a good time, guys, and
have a good evening.
Thank you. Bye.